We're the nutrient enrichment group. I'm sure you heard our names already. And we're going to focus on the concepts of nutrient enrichment we did in lab. So before we talk about enrichment, we have to talk about what a nutrient is. So essential nutrients are basically elements that you really need to grow and reproduce. And a lot of these essential elements are basically building blocks for biological processes and biological molecules. So there are basically two types that you should know. There are macromolecules, and these are molecules that you need in a very high concentration. And some of the ones that you could easily identify are like carbon and hydrogen, oxygen, nitrogen. And as you know, nitrogen is a really big part of amino acids and proteins. And macronutrients bring up a really big concept called Liebig's barrel. And this concept is basically saying how growth is limited not by all of the nutrients, but the concentration of the scarcest one. So an analogy is maybe I give you 50 pieces of bread and only 2 pieces of ham. So the most amount of ham sandwiches you can make is 2. So what you could say about the ham is that it's the primary limiting nutrient or ingredient in the sandwich. So think about the SynBio experiments, when you double the amount of phosphorus, it was oh, now you can grow. But if you double the amount of nitrogen, it's no, you need the phosphorus. So let's say I give you 700 pieces of ham, but you have the 50 pieces of bread. Well, you can still only make 25 sandwiches, unless you make very strange one bread sandwiches. So, and then you can also talk about micronutrients, and these nutrients you need in a very small concentration. And this is because after the concentration you need to grow, you're basically toxic. And as you know, you probably don't want to ingest poison because you'll die. And these organisms, they don't want to die, as a basic rule. So you try not to over-inundate these organisms with too many of these micronutrients, including iron and copper, because it's just really bad for the ecosystem, guys.
All right, so going back to the SynBio simulations that we did in lab, where we had the different phytoplankton species living in the same aquatic environment, we're going to talk about competition for a second. So competition is a mutually negative interaction between organisms, and there are 2 basic genres of competition. There's intraspecific competition, which is competition between members of the same species. And then there's interspecific competition, which is between members of different species, which is what we saw in the SynBio simulation. Within interspecific competition, we have, again, 2 types of competition. So there's interference competition, which is when one species limits the access of the other species to a limiting resource, causing the inferior species to usually go extinct. And there's exploitation competition, which is when the outcome of the entire competitive situation depends on how efficiently each species uses that limiting resource. So a species with a lower required threshold for a nutrient would be more likely to persist longer in that kind of situation. Within these competition fields, there are 2 basic possible outcomes, which are competitive exclusion, which basically means that the 2 species cannot coexist while they share the same limiting resource. Eventually, one of them will come out on top and the other one will most likely go extinct. And then there's resource partitioning, which is when those 2 species do not share the same limiting resource, so they can coexist in the same environment.
So another aspect of nutrient enrichment is the process of eutrophication. That is the natural tendency of nutrients to accumulate in large bodies of water. Previously, we learned about various nutrient cycles, such as the carbon cycle, nitrogen cycle, and phosphorus cycles, and these all contribute to the process of eutrophication. As eutrophication continues on, an ecosystem's nutrient composition can drastically change. For example, we just learned about limiting nutrients. A limiting nutrient can become so abundant that it is no longer limiting. or it can become even more scarce. It's important to realize that eutrophication is a purely natural process. However, anthropogenic nutrient input, which is basically the effects of humans on the process of eutrophication, can drastically change the surrounding environments. Essentially, man-made tools and chemicals such as fertilizers, runoff fertilizers, and pesticides can negatively impact the ecosystem and facilitate the accumulation of harmful nutrients. This can be very problematic since, as we just learned, an excess of nutrients, especially an excess of micronutrients, can become very toxic and can mean death for certain species. In some extreme cases, an excess of nutrient enrichment can lead to algal blooms, and although this burst of life may seem initially beneficial, what ends up happening is that such a large bloom of algal species tends to deplete the available amount of oxygen. This can lead to hypoxia, a lack of oxygen, and eventually the formation of dead zones, where essentially no life can exist due to the lack of oxygen. And dead zones and algal blooms are found in almost the mouths of all major river systems, so it is a very important topic of scientific discussion.
So just one last thing, if you guys notice, we also cited our sources in APA style. And the major concepts you need to know for practical are in that word cloud over there, and that's all we want to share with you guys today.
So good luck for people who have their practicals today. We know we do.